In this video, I'm going to explain the higher level genetics, specifically dihybrid inheritance and linkage. Dihybrid inheritance means the inheritance of two genes at the same time. The two genes will be situated on different chromosomes, and the chromosomes can assort independently. This means that there are plenty of possible combination of gametes and helps to illustrate how meiosis can lead to genetic variation. This shows all the possible arrangements of chromosomes in the gametes, assuming independent assortment. You can see that if there were different alleles on each of the chromosomes, you get lots of possible combinations, up to 4 in this case with two genes. When making gametes in dihybrid inheritance, you must remember that you get one of each letter in each gamete. So in this case, you would get one big A and one big B. So all of the gametes will be the same, because remember, gametes are haploid and not diploid, so you can't have two of the same letter. In this case, because you're heterozygous on the A, you're going to get two different types of gametes. You're going to have the big A, big B gamete, and you're also going to have the little a, big B gamete. This case, where you are the double heterozygote, you get the 4 possible combinations as shown in the picture previously. So you're going to have big A, big B, you're going to have big A, little b, you're going to have little a, big B, and you're going to have little a, little b. This is really important in understanding the rest of dihybrid inheritance, so it's crucial that you're confident that you can create gametes. I'm going to work through a dihybrid test cross using A and B as my example. So in this case, I'm going to have my parental genotypes of big A, big B, little b, and they're going to cross with the homozygous recessive, little a, little b. We would call this a dihybrid test cross. Because the results will tell us whether the parental, this one, is homozygous or heterozygous. So first of all, we need to create our gametes. So in the first parent, the double heterozygote, we have a big A, big B gamete, we have a big A, little b gamete, we have a little a, big B gamete, and we have a little a, little b gamete. And in this one, all of the gametes are going to be little a, little b. Where they're all the same, you can just put 1, because that then simplifies your ratio at the end. So to make my Punnett square, it's going to be a 1/4 Punnett square. So I'll have my little a, little b in there. Then big A, big B, big A, little b, little a, big B, and little a, little b. So we're going to be big A, big B, little b, big A, little b, little b, little a, big B, little b, and little a, little b, little b. So in terms of the phenotypic ratios, we have big A, big B, big A, little b, big B, and little a, little b. So that gives us a 1:1:1:1 ratio, which is the standard Mendelian ratio for a dihybrid test cross. The other standard Mendelian ratio that you need to remember for dihybrid inheritance is the result of the dihybrid heterozygous cross. So that would be big A, big B, little b crossed with big A, big B, little b, and that one is 9:3 to 3:1. Up to now, we've looked at inheritance of the two genes when they are on different chromosomes, so they can independently assort. Mendel assumed that this was always the case in his experiments, but subsequent work has shown that that's not always true. If the two genes are on the same chromosome, inheritance happens slightly differently. So in this case, if we have gene A and gene B, and then on this chromosome we're going to have the recessive, little a and little b. So the genotype of this organism is big A, big B, little b, just like we saw before. However, when we make the gametes for this particular organism, what we're going to find is that this chromosome is going to make a gamete and this chromosome is going to make a gamete. Consequently, our possible gametes are going to be big A, big B, or little a, little b. Okay? To be able to get the big A, little b gamete, or indeed the little a, big B gamete, then we are going to need to have crossing over. So we need to have a crossing over situation happening here, so that you're changing parts of the chromosomes. Now obviously this does happen, but it doesn't happen as a standard. It doesn't happen every single time, and consequently we find that these gametes, which we call the recombinant gametes, are far less common, and that has an impact on phenotypic ratio. With linkage, and that's what this is called, linkage, not to be confused with sex linkage. Sex linkage is any gene carried on a sex chromosome. Linkage, or autosomal linkage, to give it its full name, is where two genes are carried on the same chromosome. We need to notate this slightly differently, because as previously shown, if we had this genotype, we do not form all of the gametes. So we need to write it in such a way that we know that we're not dealing with this situation. So the way that we write it is actually we're on lines, so it's like it's on the chromosome. So you have the big A, big B, and then the little a, little b. And so that means that you know you're going to form a big A, big B gamete and a little b gamete. You might form some big A, little b gametes and some little a, big B gametes, but it would only be a few.